Hello, in this video we will discuss about the antigen structure, types and binding sites. So the antigen is molecule which is able to generate antibodies. So that is why it is known as the antibody generator. So the molecule generates antibodies. So the AG, antibody generator. So this is basically the different type of antigen we will discuss in this video. So first of all, here is the person which contain inside is the antigen in their own body is known as the endogenous antigen, while the bacteria, virus or some proteins which that is basically the exogenous antigen which infect the human. So that is why it is the exogenous. So here is the molecules generator antibody is also known as the AG, antibody generator. So while here is the antibodies which that is produced against this antigen. So that is why it is known as the antibodies. So the antigen, for example, foreign antigen is known as the exogenous antigen, while inside of the human is contained endogenous or self antigen. So here we will discuss about the self antigen. So the self antigen contain all cells which that is contain some antigen in the form of epitope. So this epitope is known as the antigen, is self endogenous. So it is basically recognized different types. So the T helper cell which that is recognized this antigen, for example, so the CD4 receptor containing CD4 cell which that is contain MHC class 1 molecule which that recognize after the recognition, basically after the recognition that will be produce the antibodies against our cell. So it is very destructive mechanism. So it is inhibited by the T regulatory cell which that is produce interleukin 10 and TGF beta which that is inhibit this T helper cell. Why? Because it is no need to produce antibodies against self antigen. So why? Let's begin to understand with the whole detail. So here is, for example, the CD4 cells will recognize this antigen. After the recognition, without the T regulatory cell will regulate and not produce the interleukin 10 and TGF. So it means it will be stimulate the B memory cell to produce plasma cell and B memory cell. So B cell produce plasma cell and B memory cell, which that is basically against this epitope, and antibodies will be produced and the cell will be collapsed, and it means the cell will be non-functional. For example, this is known as the autoimmune disorder. Next we will also discuss, first we will understand about the antigen. So here is the virus antigen, for example, which that is antigen generator to produce antibody AB. So this antigen antibody complex will be formed. After the complex formation, the macrophage will be engulfed. So let's begin to understand the antigen. What is antigen? So the antigen is make a protein. Antigen is made up of protein, amino acid, polypeptides or polysaccharides. For example, the sugar containing chain, polysaccharide. Poly mean many, saccharone means sugar. So here is the different type of, here is the epitope are present in the antigen, which that is this epitope is recognized by the antibodies. So this epitope is known as the antigen. So the epitope of protein antigen basically we will discuss, and we can categorized into the conformational epitope and linear epitope. So the conformational epitope is the 3D structure, is the conformation of epitope or 3D structure of epitope define its function. So its function will be different and contain many more epitope, which that is in the form of 3D structure. While on the other hand, the linear epitope means the linear amino acid sequence define its function. So it is the different function. So basically amino acid, basically here is the conformational epitope. So this is conformational epitope. So here is the linear epitope, which that is linear amino acid, which that is a sequence define its function. So it's a different function which that is in fact in the human. So this antigen basically made up of amino acid. In the linear chain is the polypeptide. So for example, here is the different type of linear epitope, which that is the epitope of protein antigen. So this is the basically leucine, glycine, alanine, and alanine, leucine, glycine, methionine, which that is the sequence, and different sequence against this sequence will be antibodies produced. On the other hand, the partial antigen and complete antigen is also we can categorized, which that is partially antigen mean haptin. Haptin is the partial antigen, while the complete antigen is able to induce immune response, while haptin is not able to induce antibody and not produce immune response. So the haptin contain a basically haptin plus carrier protein. This haptin plus carrier protein is in the form of two types, complex or simple. Polyvalent, and two or more antibodies are required for it, while the combining sites is basically and simple is the univalent. While on the other hand, it is not completely immune response will be give, but half. On the other hand, the able to induce the complete immune response is the antibody, is the foreign antibody is the basically formation, that is the evoke response, basically immune response. So the antibody here is the, for example, the MHC class 2 molecule we recognize in the presence of T cell receptor, and that basically recognize antigen, and T cell will be give the antigen presenting molecule to T cell receptor, and that will be a B cell proliferate into B memory cell and plasma cell to produce antibody and give the response. So that is the basically complete antigen, while on the other hand the partial antigen will not do that, but it will be basically two or more antibody combining sites. So there are two or more antibodies combining site. So let's begin to understand about the exogenous antigen as well as endogenous antigen. So the exogenous antigen means the outside of the body. For example, the outside of the body, which that is virus, bacteria, or protein. For example, the protein, which that is some protein foreign. So here is the antigen presenting cell. When antigen will be invade into the human, antigen presenting cell will be recognized, and after the recognition the piece of antigen will be picked, and piece of antigen will be recognized by the T helper cell. So the T helper cell is the antigen presenting cell with the MHC class 2 with the CD4 molecules of the MHC class 2 is the CD4 molecule attached, and that is the basically produce chemokine. This chemokine is basically T helper cell will be produced. After the chemokine production, the B cell proliferation and the recruitment of the B cell for the production of antibodies against this antigen, while here the B cell will be converted into the B memory cell as well as the plasma cell. Plasma cell produce the rapid antibodies against this antigen, while the B cell will be remain into the lymphatic system for the further infection, next infection, and quick response as compared to the first infection. So I hope you make sense. Let's begin to understand about the endogenous antigen inside of the body. So endogenous means the inside of the body, which that is the antigen is present. For example, our cell contain a different type of epitope. That is epitope is known as the antigen. This antigen is the different type. For example, here is the tissue, which this tissue is the pancreatic tissue. Pancreatic tissue is the islet of beta, and islet of Langerhans contain a beta cell. This beta cell is basically recognized by the antigen presenting cell, dendritic cell, to HLA class 2 molecule with the antigen presenting cell with the MHC class 2 molecule of the CD4 T cell. After the CD4 T cell recognition, the CD4 molecule with the attachment to produce the give the response, but the one major thing is that the T regulatory cell will be inhibit this mechanism with the attachment of PDI molecule or PDI receptor with the CD4 T cell molecule, and produce the interleukin 10 and TGF beta, which that is basically inhibit the reaction and recruitment of the CD8 plus T cell. Why? Because CD8 plus T cell will kill the tissue of the pancreas, for example. So that is known as the autoimmune disorder. But when the positive effect due to the interleukin 2 production to recruit the CD8 T cell after the recognition, and that means it will be recognized and kill the body cell, islet of Langerhans tissue, and HLA class 1 molecule, which that is represent the antigen of the beta cell. So that's beta cell will be recognized. After this recognition, so the CD8 T cell will be kill that islet of Langerhans. So it means the cell lysis and produce the granzyme as well as perforin. So the perforin or perforin, you can see. So here is my mistake about that. So the Grange enzyme and perforin. This perforin is the special protein which that creates the pores. So that is also known as the porin. So basically it's kill the body cell. For example, this is in the form of autoimmune disorder, myasthenia gravis, sickle cell anemia, and diabetes mellitus 1, which that is the character of the autoimmune disorder. But the T regulatory cell will be inhibit this mechanism and produce the interleukin 10 and TGF beta, which that is inhibit the CD4 plus T cell to recognize this antigen, which that's come from the dendritic cell, which that is recognize it. So on the other hand, the antigen of antibodies, which that is also produced. For example, it is antigen and antibody, for example, and when inject these antibodies, it will become a antigen. Why? Because it's contain a epitope. Against this epitope, antibodies produced in the human, so that is known as the exogenous antigen, play important role as an exogenous antigen, and secondary antibody will be bound with the epitope of these antibodies, and these secondary antibodies is known as the antibody, while the primary antibodies is known as the antigen. So the against antigen will be produced, and that means it is the secondary antibodies. While on the other hand, one more thing is important, that for example, the tissue is a damaged or infected tissue, basically infected tissue with the infection of the cancer or viral infection. So the CD8 T cell will be recognized by the CD8 molecule with the MHC class 1 by the representation of the antigen presenting molecule will represent, and after the representation, CD8 T cell will be produce cytokines. These cytokines proliferate naive T cell proliferation to produce CD8 T cell. This CD8 T cell is give the positive effect and more recruitment of the CD8 to kill the more infected cells. So that's also produce grain enzyme and grain enzyme as well as perforin, which that is kill the bursting of the cell. So that means the apoptosis will be occur. So I hope you make sense about these things. So you can see. So this is production of the CD8 T cell, more proliferation to kill more that infected cell. So thanks for watching. Please make sure to subscribe, like and share to your friends, and okay, bye.